Right. Hello and welcome to another video. This week we are going to be discussing herbal therapeutics for Alzheimer's disease, ancient Indian medicine system for the modern viewpoint. This paper was written by Shikha Kushwah, Neha Maurya, Sandeep Kushwaha, Luciana Scotti, Aakash Chawade, and Ashutosh. This is in general Bentham science. So in this article, basically the authors are going to give some reasons as to why certain herbal plants may be beneficial in treating Alzheimer's disease. These plants include turmeric, ginkgo, ashwagandha, shank, pushpi, giloy, gorukola, garlic, tulsi, ginger, and cinnamon. And to kind of preface this, if you haven't seen any of my other videos, Alzheimer's disease is basically a type of neurodegenerative disease characterized by the buildup of a type of protein known as amyloid plaque. And this protein essentially kind of builds up in the brain and eventually starts causing downstream actions such as blocking neurons from talking to each other, and essentially leads to a cascading effect that will essentially lead to widespread neurodegeneration across the brain and lead to memory loss, cognitive dysfunction, sometimes depression, brain disorders, and inability to just do activities of daily living, and thus requiring some sort of help from other people. And usually, in most of the cases, people who have this disease tend to die. These authors have come up with some new statistics as to how much this disease kind of impacts the world. So in Europe, the combined incidence rate for AD among those 65 and older was 19.4. This just means that about 1.94% of people are kind of, when they are 65 and older, will get this disease. And in the US here, it says it's about 15 per 1,000 person years, about 1.5%. So we have some FDA approved drugs that are not very they don't do the work very well. These are acetylcholinesterase inhibitors and NMDA receptor antagonists. But these authors are kind of positioning that there might be some other ways to treat these diseases. So the first one that we will be looking at is turmeric, or also known as the spice of life. So turmeric seems to exhibit healing antioxidant, anti-inflammatory, anti-cancer, and anti-amyloid properties. This was something that was used during Vedic times. Vedic times is basically a time in India where it kind of, I guess you can compare it to the Renaissance in Europe, but this happened in India where the kind of the cultural the cultural uh, sphere kind of developed and grew in a way that incorporated different ideas and methods from different cultures from different areas. This is when India was kind of becoming like a its own sort of very modern civilization based on that time. So it kind of came together, and this usually in this time it tends to be known as the Vedic times or the Ayurvedic times. So what this author found was that its medicinal properties of turmeric have been documented to be used in the Ayurveda, Siddha, and Unani systems of medicine. And tau and the accumulation of AB is the primary hallmark of Alzheimer's disease, right? Curcumin, which is it's a prime product in turmeric, has been shown to essentially focuses on the two histology indicators. So tau and AB, studies have found that curcumin doses ranging from zero point 0.1 to 1.0 moles. So the doses of 0.1 to 1.0 moles have disaggregated fibrillar AB40, inhibited AB40 aggregation inside the brain, and prevented development of AB42. So AB40 is usually kind of the soluble version of amyloid, and AB42 is the insoluble type, which is the one that forms a lot of oligomers. So AB40 is not usually bad, but AB42 is the bad one. The fact that it's able to kind of stop this is pretty impressive. Another thing to know is that most of these studies were done in transgenic mice, that mice models that are kind of groomed to kind of exhibit a specific disease when they grow older. And in these mice, these were Alzheimer's transgenic mice, and they showed the expression of protein inflammatory cytokine IL-1 and astrocytic inflammatory marker GFAP, which was significantly reduced at 160 parts per million dose of curcumin, which is the active ingredient in turmeric. The low dose of curcumin reduces the production of amyloid beta plaques and the oligomerization tendency for amyloid beta formation in transgenic 2576 mice brains. Another important thing to take into account here is that The bioavailability of curcumin is not that great, but these researchers were able to enhance the bioavailability and solubility of curcumin by using nanoparticles, and they found that when they used the nanoparticles, it exhibited improved curcumin transport towards the brain within 24 hours. And if it's able to kind of stay in the brain longer, it's able to do this. So we have amyloid precursor. This is a good graph that will kind of explain this better. So amyloid precursor protein comes in. Amyloid beta secretase is a type of enzyme that cleaves it, and then it cleaves it to 40 or 42. Usually, amyloid beta 42 is one that aggregates more. Amyloid 40s it aggregates, but it's soluble, so it's able to be cleared out easily by the CSF. But this cannot be cleared out. So when AB42 is not cleared out, it becomes oligomeric, forms oligomers, and then it becomes an amyloid plaque, causing neuronal death. Curcumin, active product, can help clearance of plaque, which that comes by clearing the oligomers. It can also form anti-inflammatory response by blocking the inflammatory response that comes from amyloid plaque formation. And it can also inhibit the tau hypophosphorylation. That if you look at my other videos, I'll kind of explain this what tau hypophosphorylation is. But curcumin is able to stop that, and it also helps in tangle clearance, which is kind of like a multi-shot emphasis on this thing. So which is that's a good thing. The fact that it's able to do that is kind of impressive. Another one is ginkgo biloba. This has an active ingredient known as EGB761. Let's just talk about the extract. EGB761 obtained from this plant helps in enhancing antioxidant activity in the brain cells, which is also free radical scavenging. This means that it's able to stop the rapid oxidation that happens when amyloid plaques come in, and it blocks that from causing further damage. It's able to stop cytochrome C, which is a mitochondrial protein that is usually released when cell death has been planned for the cell specifically. So it comes in and it stops this from happening, and that kind of reduces the amount of free radicals that can be produced from that, and also kind of helps the cell maintain itself so that other things can come in and help it maintain and kind of if it's bruised or not or needs to be healed, these things can come and help the neuron heal itself better. The active phytochemical compound present in ginkgo biloba extract prevents AB aggregation and amyloidogenesis. That means it stops the generation of further amyloid protein, so you will clear it out as well. Here is just a little graph of what ginkgo biloba is able to do. So what is it able to do? It is able to increase neuroplasticity, and it's also neuroprotective and has a neuroregeneration effect. It's able to increase the accumulation of macroglia around amyloid plaques. Macroglia are basically the immune cells that are only found within the CNS, the central nervous system. It decreases the pro-inflammatory cytokines IL-1, IL-6, TNF alpha. And if you know anything about neuroscience, these are the worst kind of, or biology in general, these are the kind of the worst type of cytokines that you want to have in the body. So it reduces these pro-inflammatory and increases anti-inflammatory cytokines, which is IL-4, IL-13, and tumor growth factor beta. This ginkgo biloba extract also has the capacity to scavenge reactive oxygen species, increases or helps the body increase the endogenous superoxide dismutase or glutathione reductase, which are basically antioxidant species, reduces lipid peroxidation, which produces reactive oxygen species, and it also reduces nitric oxide synthesis. It also has the capacity to produce or to increase expression of genes encoding antioxidants. So it's very good with antioxidants. Lastly, it will decrease reactive oxygen species production, as I've already stated, as well as mitochondrial DNA protection. It seems to protect it, protection against amyloid beta, and it would increase the availability of ATP. So if the cell has been damaged or injured from the cascading effect of amyloid beta, this extract seems to come in and kind of help the brain heal and stops further action from going into, stops further degeneration from occurring. So this is just several mechanisms of this. The other one that was discussed in this paper was Withania somnifera, or also known as ashwagandha. So what we need to know about ashwagandha is that it comes in certain varieties and certain forms. So there's extracts that come from this. The extracts of WS have the potential to maintain a healthy immune system. The ergostein type steroidal lactones, we have wethasominiferin A, dehydrowithanolide R, withasominide, withasomidedienone, withaferrin A, as shown in figure 6, withasominophorals A to C, phytosterol, cytoendosides 7 to 10, and beta cytosterol are among the steroidal chemicals found in Withonia spinifera extracts. So, the reason why I say all this is I'll explain in a second why it's important, why all these varieties are important in helping cure this disease. So, withasominophorals A is a withanolide obtained from the roots of this plant, ashwagandha. It has been proven to counteract amyloid beta 42 induced toxicity in human neurons, and it's been seen here that withanamides A to C uh, seems to stop within the amyloid beta protein there are motifs which are basically amino acid residues. So what this thing does is that it suppresses these residues from 25 to 35, and when it suppresses them, it stops the oligomerization of amyloid beta. So what this means is that it just suppresses fiber formation. It stops the oligomerization of amyloid beta. And it was found that axons and dendrites of neurons were greatly regenerated after continuous treatment with the methanol extract of ashwagandha. Additionally, cellular regeneration, ashwagandha improved mice's memory by reversing amyloid peptide induced memory loss, and some studies have proven that oral administration of Withania somnifera extract contained withanolides and withanosides have halted the progression of AD. It also recovers plaque pathology and the development of AB peptides and oligomers in the brains of middle age and old age APP presenilin 1 transgenic mice. Okay, that was a lot. But what is this saying basically? So, APP presenilin 1 transgenic mice are a type of mouse um, uh, genealogy or line that is specifically bred to exhibit a type of Alzheimer's disease that is commonly known as familial Alzheimer's disease. People who have this have a very low capability to clear out amyloid beta, and they tend to develop amyloid beta accumulation Alzheimer's disease at a very young age. And these researchers found that this specific extract is able to stop that, but only in mice that have been bred to kind of exhibit this type of phenotype. So the next one that we're going to be discussing is shank pushpi. Keep in mind that most of these plants are kind of found in India, so they have the native names that are specific to them. Shank shupi is a type of plant, and there are about four varieties of this. We have Convolvulus pluriculus, Choisy, Clitoria ternatia lin, Canscora decusata schult, Evolvus alcinoides lin, and these are considered as sources for Shankshruti by Indian practitioners. This is a nervine tonic for improving cognitive and memory function, or more specifically, it kind of helps with anti-stress and memory. Okay, so there are metabolites that come from this. We have flavanol glycosides, triterpenoids, steroids, and anthocyanins. These have been identified as principal bioactive compounds of the Shank-Shupi's acid extract, and compounds can be responsible for various pharmacological effects in addition to enhancing cognitive ability. So let's begin. Ethanolic extract of Shank-Shupi enhances the antioxidant activity at a significant level inside brain cells. This is only in vitro. It scavenges free radicals and acts as an antioxidant. Recent studies shows that it lowers amyloid beta accumulation in the brain, which protects against memory loss in Alzheimer's disease. So, neuropharmacological based activity also indicates that this E-alsonidase, which is a variety of the Shankshupi, is the most effective of the four types and boosts antioxidant enzyme activity and controls tau-induced oxidative stress. This includes acetylcholine esterase activity. This is an improvement in passive avoidance. This can be shown as it can be shown outwardly as the improvement of passive avoidance learning in young adults, and this has also been shown to only work in this has only worked in mice that were kind of given this in a in when they're infants. So when they're incubated with this is how this only worked if they were incubated with this or were babies and were given this root when they were babies. These effects seem to only work from that perspective. So the other root here that or leaf that was found to show some activity to reduce Alzheimer's disease was Tinospora cordifolia, or known as giloy. So this has been utilized in Ayurvedic medicine. It is referred to as an adaptogen or rejuvenator. Has antioxidant, immunomodulatory, anti-inflammatory, anti-hyperglycemic, antispasmodic, anti-ulcer, and many other properties. The putative anti-stress benefits of Tinospora cordifolia, or I'm just going to call it TC, have recently come to light in studies involving both humans and animals. So here we see so the most plausible antidepressant mechanisms involve the inhibition of amines inside the brain. So this means that it has antidepressant effects by blocking amines such as serotonin, dopamine from kind of getting reabsorbed back into the brain, keeping them in in the synaptic space longer, and kind of having some antidepressant effects in that way. Several research groups are working towards protection against neuroinflammation and oxidative stress. Different antioxidant compounds have been studied till now to analyze the potential for protection against neuroinflammation and oxidative stress, as they're responsible for altered mitochondrial activity and production of free radicals. So they're trying to see if this is able to help with this, with the altered mitochondrial activity and reduction of stress or neuroinflammation and oxidative stress causing activity. So this here it says a recent study of an antioxidant and anti-inflammatory effect of T. cordifolia extract was able to attenuate nuclear factor kappa B translocation, which is a type of it's a type of molecule that kind of cascades into becoming it it causes usually downstream effects that would lead to the death of a neuron. So it kind of attenuates it and makes it so that it doesn't lead to that. Um, LPS activated RAW2647 cells are or aqueous extract of T. cardifolia dramatically decreased gene expression of inflammatory cytokines like IL-1, TNF alpha, hepcidin, as well as nitrous oxide production. Tin- Tinosporicide was detected by HBLC analysis, which is likely to have contributed to T. cordifolia's ability to reduce inflammation. The next one here we have is known as Gotu Cola or Centella asiatica. Seems to be a plant that's found mostly in Asia and utilized as a traditional medicine for more than 2,000 years. Okay, so the whole plant extract, including ethanolic aqueous extract or C. asiatica, have both been discovered to possess a multitude of therapeutic properties, as well as as well as biological activities. So, what are these activities? According to Ayurveda, this plant is for neurons and other brain cells and can boost intelligence, memory. Its compounds were discovered to have a protective effect against AB-induced neurotoxicity. It's linked to AD, which is linked to AD dementia, has antioxidative defense system in cells, such as glutathione reductase increase, glutathione peroxidase, and levels of glutathione disulfide and glutathione is modulated by CAE. It aids in the death of AB cells in vitro, making it a promising medicine in the treatment and prevention of Alzheimer's disease. Studies have shown that C. asiatica can treat Alzheimer's disease like disease in rats by inhibiting hyperphosphorylated tau. That's pretty good. And biosynthetic and anti-apoptosis protein. All right, so moving on. The next one here is alum sativum, or mostly known as garlic. We know what garlic is essentially, so it's a belongs in the Amaryllidaceae family and utilized as a herbal medicine since ancient times. Aged garlic extract, or AGE, is a non-toxic solvent extract of garlic powder. It is derived from an extended extraction period of up to 15 months at room temperature. Or so we could say more than or up to 15 months. Less than or up to 15 months at room temperature. The aging process converts allicin, an unstable compound, is transformed into a stable substance. It contains two key compounds, D-allyl disulfide and S-allyl cysteine, as shown as in figure 10a-b. Additional phytochemicals found in AGE include alexine, ajoine, polyphenols, thiosulfonates, and flavonoids. Recently, there have been some attenuation to the possibility of AGE to improve cognitive impairment in Alzheimer's disease. Amyloid beta induces the expression of GRP78, potentiates the ER stress induced neuronal death. The SAC component of AGE was found to have a strong neuroprotective impact on ER stress induced neuronal death. All right, so they basically found that AGE has showed progressive degradation of hippocampal based slowed progressive degradation of hippocampal based cognitive activities by reducing the accumulation of amyloid beta, and cognitive impairment is a typical pathology in AD, and AGE has greatly reduced cognitive impairment in rats by enhancing working memory and reference memory. So it seems that fresh garlic extract can kind of reduce amyloid beta fibrils after 2 to 3 days of incubation. The highest defibrillation was detected. The anti-amyloidogenic activity of the boiled aqueous garlic extract was retained, indicating that the anti-amyloidogenic activity of the garlic extract is not enzymatic in nature. As a result, allium sativum, potentially a helpful substance that can be utilized to treat AD as shown in figure 11. Ingesting garlic on a daily basis may also suppress the accumulation of AB inside the human brain. That's probably the most important thing you take. So if you can kind of eat this as often as you can potentially kind of reduce amyloid beta formation. All right, so here we have the next one. I think this is the second last one. Ossimum sanctum belongs to the Lamicae family and has been utilized in Ayurveda since ancient times due to its diverse healing properties. Considered an incomparable one herb of India and has been cherished for its healing capacity. The chemical composition of O sanctum is intricate and includes numerous nutrients and biologically active compounds. There are numerous biological active phytochemicals present in holy basil stem and leaves, including um, terpenoids, saponoids, tannins, and flavonoids. The phenolic group that contains active compounds exhibits anti-inflammatory and antioxidant activities are apigenin, figure 12A, rosmarinic acid, figure 12B, acimethicin. So where's 12A and 12B? So this is 12A, that's what it looks like, and this is 12B, rosmarinic acid. Seem to be the active product. Hypercholesteremia induced erythrocyte lipid peroxidation activity was inhibited in male albino rabbits, treating them with an aqueous extract of O sanctum in a dose dependent manner. Additionally, oral feeding significantly reduces peroxidative damage induced by hypercholesteremia in the liver and aortic tissue. The compounds have been extracted from O sanctum aqueous extract are civil-mestin eugenol, civicillinol, and apigenin, which are studied for their cyclooxygenase inhibitory activity or anti-inflammatory activity. Study was conducted to analyze the potential of O-sanctum extract as an anti-amnesic and nootropic agent in mice. Amnesic response of mice towards scopolamine, so 0.04 milligram per kilogram, diazepam 1 milligram per kilogram, and aging were reduced by an aqueous extract by an entire plant. O sanctum, the comparison of control piracetam treated scopolamine and elderly groups of mice have shown a significant lower transfer latency and increased step down latency when treated with O sanctum extract. So preparation of O sanctum might be useful for treating cognitive disorders including Alzheimer's disease and dementia. Production of choline acetyl and acetyltransferase can be induced and maintained in human cerebral microvascular endothelial cells. In an ethanolic extract from O sanctum, it may be a candidate for neuroproductive substances, but a lot of in vitro research is still required in human cerebral microvascular endothelial cells. An ethanolic extract from O sanctum can induce and sustain the production of choline acetyltransferase. Thus, it can be a candidate for neuroprotective substance, but further in vitro studies are needed. So, acetyltransferase is essentially it's basically the precursor to acetyltransfer to the cholinesterase. So this enzyme would kind of produce as a um, cholinesterase. So the fact that this thing is able to is able to help this grow is quite good, which explains why it's neuroprotective. All right, so here we have the next one. Here is ginger. It's been utilized in as in Indian spices. Inflammation plays a key role in etiology of AD. So anti-inflammatory drugs for the treatment of the disease. A recent study discovered that a mixture of ginger extract and Zingiber officinal, Roscoe, and Alpinia galanga can prevent THP1 cells from being activated by tumor necrosis factor, IL-1, LPS, and 8 amyloid beta. Molecular dynamic simulation base study of ginger's two components referred to as MOL1 and MOL2 were identified as potential natural inhibitors of HSS-ACHE, acetylcholinesterase, which is the enzyme that kills acetylcholine. And it was proved to be just as effective as the current medicine of choice, which is pretty interesting. Huh. The suppression of many inflammatory response markers by GE shows that this herbal preparation could be a promising drug for AD. Many interesting pharmacological and physiological functions of gingerols and sugar oils have been documented, including antipyretic, cardiotonic, chemopreventative, anti-inflammatory, and oxidant capabilities. So this is a bioactive chemical known as gingerol. Good to take into note that this is the mixture that was used in the study: Zingbar officinal and Alpinia galanga. All right, so the next one in this study is cinnamon, which is very common. And cinnamon has an active ingredient. So far, the main phenolic chemicals isolated from cinnamon are EGCG or epigallocatechin, as shown in figure 14a. So this is what it looks like. This is catechin that can cross the blood-brain barrier and can target all three Alzheimer's disease hallmarks. So it inhibits aggregation of amyloid beta, inhibition of tau hyperphosphorylation, and degrades plaques, so it stops plaques from forming. Cinnamon reduces the production, accumulation, and toxicity of amyloid beta plaques in PC12 neuronal cells. In AD fly models, the inhibitory effects are similar. It also aids in tau phosphorylation inhibition and acetylcholinesterase activity suppression. It inhibits the generation of intracellular reactive oxygen species and the expression of pro-inflammatory cytokines such as NFKB, IL-6, and TNF. Last one here is Azadirachta indica, which is an evergreen tea found throughout India and neighboring countries. Neem is derived from the Sanskrit word nimba, which means nature's drugstore or panacea. More than 300 chemically diverse phytochemicals have been isolated from neem. The major phytochemicals present in neem are glycoproteins, aldehydes. Experimental study on rats performed by Mighty et al. Increased the number of ambulations comparable to diazepam. The study also suggests that antidepressant activity of this medicinal plant. In another experiment, limanoids extracted from the neem fruits decreased the extensive aggregation of tau in vitro and led to the formation of thin, short, and fragile aggregates. Limanoids reverted tau-mediated toxicity at 1 micromolar concentration and were also able to overcome oxidative stress. As this would highlight exactly what the list of various etiological hypotheses and their characteristic features related with Alzheimer's disease. So we have AB cascade, AB peptides deposit as senile plaques, intraneuronal neurofibrillary tangles / neurodegeneration. We have the tau hypothesis, which is new. This is basic. So essentially, this is the hypothesis of how Alzheimer's disease um, is generated. We have the amyloid beta cascade, which is the amyloid beta proteins I discussed earlier, forming plaques and in between neurons and inhibiting function from those neurons and leading to a cascading effect. We have the tau hypothesis, which is the formation of neurofibrillary tangles within neurons, causing death of neurons within themselves. And then we have the inflammation hypothesis, which is the reactive gliosis and neuroinflammation, which comes from a lot of degeneration. So when neurons start to fail, they produce reactive oxygen species, and these reactive oxygen species go on to cause a lot of inflammation. And the inflammation is triggers microglia. Microglia come in and cause even more inflammation. And then we have the cholinergic and oxidative stress hypothesis, which is that cholinergic neurons are damaged, and then this leads to cellular oxidative stress. So, what is the conclusion for this paper? Alzheimer's disease is a global health concern, and it's rising. A number of evidence collected through clinical, animal, and in vitro studies indicates that herbal plants reviewed in this research help in neurogenesis and have many other therapeutic benefits. Traditional medicines with a strong knowledge base combined with modern science and techniques may improve the formulations of creating drugs for this disease. And it's important to also note that most of these studies were just done in rats, and the neurons that were tested usually were in vitro. So there's a lot more that needs to be taken into account before we can try to put this or try to state this or to to make some causative or make some connections within these specific studies as to how they can cause the treatment of Alzheimer's disease, which isn't necessarily what these authors are saying. The plants are not going to cause the treatment of the disease, but it's an avenue or a way of which we can start looking at the treatments. We can stop focusing on chemical derived drugs. We can look at plant or naturopathic ways of or forming of treatments for this disease, and also using other ways such as using drugs as well as plants to kind of come up with a multimodal way of treating this disease, because it's as you can see there's a lot of effects that come from neurons dying. And so you have the amyloid beta hypothesis, which states that the amyloid plaque is going to form. The amyloid plaque is going to come and cause widespread downstream effects for neurons and making those neurons incapable of communicating with each other. The neurons start dying, and when the neurons start dying, they will release harmful cytokines, and that causes further inflammation. The inflammatory cytokines will signal the immune system, which is the microglia, to come and collect it. And when the new and microglia arrive, they usually would kind of start releasing even more cytokines, and those cytokines will further go to healthy neurons that have not even been affected by the amyloid plaque, and those neurons will start dying, and the effect will just go furthermore. And the fact is, this is something that is going to get worse, and if we don't look into treating it right away, it might be very difficult. All right, thanks for watching, and take care.